The story of DNA's secret life, chromatin and its magical helpers. Once upon a time, inside every tiny cell of our body, there lived a long and twisty character named DNA. DNA was not just any long thread, it carried the instructions for life itself. But there was one big problem: DNA was incredibly long. If you stretched out the DNA from just one cell, it would measure almost 2 meters. Imagine trying to fit 2 meters of string into a space so tiny that it could only be seen under a powerful microscope. DNA needed a clever way to stay compact, neat, and protected inside the nucleus, the little home at the center of the cell. To solve this problem, DNA found friends, proteins called histones. These histones were like little spools, and DNA wrapped itself around them, creating structures called nucleosomes. If you picture beads on a string, you get the right image. DNA loops around histones twice, forming neat packages that can be further coiled and twisted. These packages make up chromatin, the material inside the nucleus that allows DNA to be stored without tangling. And to hold everything together even tighter, another protein called histone H1 comes in, acting like a clamp that locks the DNA onto the nucleosome. This creates stability and order. But there was more to the story. DNA was not just about being stored, it also had to be read. Imagine if a library had all its books tightly locked in boxes. No one could read them, and the knowledge would be wasted. Cells faced a similar challenge: how to balance keeping DNA safe and organized with making sure that genes could be accessed when needed. The solution lay in different types of chromatin. Some chromatin stayed very tightly packed, like books locked away. This was called heterochromatin. Under a microscope, heterochromatin looked darker and denser. It was like a hidden library section where information was hard to access. Genes in heterochromatin were usually turned off because the DNA was wrapped too tightly to be read. Scientists sometimes described heterochromatin as silent DNA. Interestingly, this silent state was not useless. It was necessary for controlling which genes were active and for keeping repetitive DNA sequences quiet. Even the inactive X chromosome in females, called a bar body, showed up as heterochromatin on the edge of the nucleus. On the other side, there was euchromatin. This was the more relaxed, lighter version of chromatin. Euchromatin was like an open bookshelf where books were available to be read. Genes located in euchromatin were active, accessible, and ready to be transcribed into RNA, which would then guide protein production. Cells used euchromatin when they needed to respond quickly and make proteins for growth, repair, or function. The difference between heterochromatin and euchromatin was like the difference between a closed, locked book and an open one on your desk. Now, what determined whether DNA was locked away in heterochromatin or freely available in euchromatin? This is where chemical signals came into the story. DNA and histones could be modified, almost like being tagged with notes telling the cell what to do. These modifications acted like switches and dimmers, turning genes on or off or adjusting how tightly DNA was wound. 3 major players in this chemical story were DNA methylation, histone methylation, and histone acetylation, with histone deacetylation as the opposite of acetylation. DNA methylation was like stamping a little chemical mark, called a methyl group, onto the DNA itself. These marks usually landed on places in the DNA that were rich in cytosine, followed by guanine, known as CpG islands. When DNA was methylated in these areas, it was like placing a "do not disturb" sign on a gene. The gene would stay quiet, silenced by the methyl mark. This process was crucial for many things: aging, controlling transposable elements, the wandering, jumping genes in our DNA, genomic imprinting where only one copy of a gene from either the mother or father is used, and preventing diseases. But when DNA methylation went wrong, trouble followed. Too much or too little methylation could silence important protective genes or activate harmful ones, contributing to cancer and other conditions. In Fragile X syndrome, for instance, abnormal DNA methylation disrupts gene expression, leading to disease. Histones, too, could be chemically modified. One way was histone methylation. This involved adding methyl groups to histones, especially on certain amino acids like lysine and arginine. Depending on where the methyl groups were attached, histone methylation could either silence genes or sometimes activate them. Most of the time, however, histone methylation made DNA pack more tightly, creating a closed book and reducing gene activity. Think of it as taping a book shut. Unless the tape was cut, no one could read it. Another important modification was histone acetylation. This was like loosening the grip of DNA around histones. By adding acetyl groups, the positive charges on histones were reduced, weakening their hold on negatively charged DNA. This relaxed structure allowed the DNA to spread out, turning closed chromatin into open chromatin. Suddenly the genes could be accessed, and transcription could begin. In this way, histone acetylation acted like opening the covers of books, making them available for reading. Interestingly, even hormones like thyroid hormone worked through this mechanism, activating genes by acetylating histones. But just as there was a way to loosen DNA, there was also a way to tighten it again. Histone deacetylation removed those acetyl groups, restoring the strong grip between histones and DNA. The chromatin coiled back tightly, and genes were shut off. Histone deacetylation was like closing the books and putting them back on a high shelf where no one could reach them. Scientists have discovered that in diseases such as Huntington's disease, abnormal histone deacetylation can play a role, shutting down necessary genes and contributing to symptoms. All of these processes, methylation, acetylation, deacetylation, formed what scientists now call the epigenetic code. Unlike mutations in DNA, which permanently altered the genetic script, epigenetic changes were reversible. They did not change the letters of DNA but instead changed how the letters were read. It was as if the same library could host very different stories depending on which books were opened and which stayed closed. And so, inside each cell, chromatin was never static. It shifted and adapted, opening here, closing there, responding to signals from within the body and from the outside world. During mitosis, for example, DNA had to condense completely into chromosomes so it could be divided equally into daughter cells. At other times, such as during the S phase of the cell cycle, chromatin loosened so DNA could be copied. Mitochondria, the powerhouses of the cell, had their own DNA that did not use histones, showing how versatile life storage systems could be. When you step back and look at the whole picture, the story of chromatin is not just about packaging DNA, it is about balance. The balance between storage and accessibility, silence and activity, protection and expression. Without this balance, cells would not be able to control which genes were active at which times, and life itself would fall into chaos. Think of it this way. Your DNA is like the grand library of your existence. Chromatin is the library's shelving system, keeping books safe but also allowing the right ones to be pulled out when needed. Histones are the spools and shelves, keeping the library organized. Methylation and acetylation are the sticky notes and locks that tell the librarians which books to lend out and which to keep hidden. Together, they ensure that the library runs smoothly and that the story of life continues with precision and order. The most fascinating part is that this story is not fixed. Just like a library might reorganize its books, cells can change their chromatin states in response to environment, nutrition, stress, and signals from other cells. In fact, the way you live your life, what you eat, how you sleep, the stress you carry, can influence your epigenetic marks. This means the story of chromatin is also your story, one that keeps unfolding as you grow, learn, and change. In the end, the tale of chromatin reminds us that life's secrets are not only written in the letters of DNA, but also in how those letters are folded, marked, and read. It is a story of order hidden in complexity, of silence and sound working together, of books sometimes closed but never forgotten, waiting to be opened at the right time. Every cell carries this library, and every day it tells the story that makes you, you.